Okay, hi everybody. Thank you for having me. I'm a new member of this network. Then thank you very much. And today I would like to talk to you about the endogenous factors of tree growth because we are talking a lot about tree growth and they are there and they are pretty important. And you will see why I call them the submerged part of the iceberg. Then I would like to start introducing a bit the main framework of my research, which is investigating tree growth using modeling. What a surprise! And what do I do? It's like, how can I draft a functional tree, like with the very meaningful traits that will provide me very meaningful information about how to do that, how to feed my models with quantitative information. And this is a bit what do I do. And I've been working a lot with secondary growth because, as you know, it is very important and it is the thing about wood productivity and forestry resilience. And I've been using different scales to study this growth. And the one that it is most commonly used, it's inter-annual scale with ring width and ring density. These are the two main proxies that are used to study tree growth, and they will provide information about interannual variability in tree growth. But we might be interested in knowing what happens during the growing season, then the intra-annual variability of tree grounds, because we know that especially in cold forests, growing season occurs in very specific moments of the year. And what happens in that specific moment of the year will affect tree growth. Then this is a dimension that to a scale that we can investigate with wood anatomy and with microdensity, like the very high resolution profile of density across a tree ring. But you can look further, and this is like my scale. It's the scale of physiology and cell development. If you want to know how a tree grows, then the process underlying the tree growth, and which is related to the temporal dynamics of xylem cell development into the tree ring, which change during the growing season. And then, actually, if you look at these three different scales, you are actually answered three different questions, which are all equally important about when something happened, what is happening, how an environmental factor, for instance, is affecting the capacity of a tree to conduct water into the xylem cells or to produce secondary wall thickness that will provide that will change wood mechanical properties. And if you are interested in knowing how something happens, you can look cell development, which is a very interesting scale. But luckily, the knowledge available on these three scales is not the same. And it's, of course, for historical reason, because we started studying ring width and we are now studying cell development in tree, which makes things a bit more complicated, is that a lot of information about tree growth are recovered from remote areas. This is always related to the fact that we were used to study a lot tree rings, and we were studying tree rings in places in which there were limiting factors to growth. And then, are they really representative of tree growth in general, especially when you are working with black spruce, a species that has a very large distribution area and which has been demonstrated to be little responsive to short environmental fluctuations. Then here's the thing. What I'm trying to do is just try convincing people that maybe instead of looking at a tree ring and then thinking about cell anatomy like a consequence of tree ring, and then uh, just at the bottom of the scale thinking about physiology, like when we don't know how to explain tree growth, I would like to switch the pyramid and makes people thinking about the fact that physiology and cell development is based of how a tree is growing, and then using a process-based modeling approach to study tree growth. And my task was pretty much establishing causal relationship between cell development and microdensity to provide this quantitative knowledge for fitting models with this information. And today I will I will share with you just one study I did with Hubert, which was my former PhD director. Maybe someone of you know him. And me and Sergio were my co-directors during my PhD, and Philip Rosenberg, who was is an expert in microdensity in Irae Orléans. And with this study, I had to test a conceptual framework which was designed to quantify direct and indirect relationships between environmental factors, wood formation dynamics, and filtrates, explaining microdensity. Then how did I do that? I have a latitudinal gradient, 5 sites of black spruce stands, and I've been extremely lucky because I've worked with the longest xylogenetic monitoring in the world, which was 15 years of microgores. You can see a microgore here. It's a very tiny piece of wood, which is collected each week. Then you have people bringing it at the lab and cutting it with embedding it in paraffin, cutting it with a microdome, and then counting cells at each developmental stage. And I did some measure of wood anatomy, which at the time was my specialty for substrates, and I did also measurements for microdensity with X-rays. This thanks to Philippe. And for each site, I add weather station. Then finally, I produced a dataset in which I computed for each tree ring. I computed the number of cells, and for each cell, I had the duration of the developmental dynamics, the cell traits related, diameter, wall thickness, and I was able to compute the very environmental factors that were experienced by each cell at each moment in which at each day it were developing, related and I related it to the microdensity. Then I had this very complete information. And I had a lot of variables. Then I did a PCA, which is the most straightforward thing to do. And with my surprise, there was no patterns in the sites. You can see the sites here, different colors from the colder to the warmer, and you can see that they are all mixed. But something very interesting I mentioned was that microdensity was very close to the developmental dynamics of my cells, even closer than the environmental factors that were supposed to be the ones that the cell was experiencing during its development. Then, how can we explain that better? We can do a model, a structural equation model. It's a system of regression in which you can establish direct and indirect relationships between the variables. You can see here, we make something that we call like an hypothetical model, which is based on literature and that helps us disentangling all the quantitative relationships. And what did I found? I found that there is a portion of variability of microdensity that is explained by direct relationships between environmental factor and microdensity. This is because there is physiology in there, and not everything is related to cell anatomy. You have sugars moving, and that will respond to photoperiods, so water content, and mean temperature. And you have also a portion of variability that is explained by indirect relationship with density. Then environmental factors affecting root anatomy, and finally affecting microdensity. And finally, the very important thing here was that the relationship between duration of developmental dynamics and microdensity was indirect. And this is probably related to carbon partitioning and to the way in which these developmental dynamics are driven by the carbon partitioning between the entire plants. Then finally, for doing like the main findings of this research was that plastic adjustments in wood formation can override climate responses and allow for black spruce conservative strategy. And this is because wood formation dynamics can integrate short and long-term growth signals. And for this, they have a very strong predictive power for for predicting wood density and carbon allocation across the tree ring. And finally, we should stop considering that wood anatomy is a representation, a quantitative representation of microdensity because there are non-additive effects of environmental variation on microdensity. And then microdensity display different ecological correlations when compared to cell anatomy. Then this is the ending of my presentation. I would like to thank you. I would like to thank all the my collaborators and looking forward to explore a bit more about my iceberg with other proxies that I was not able to consider in these studies. And I'm here if you have any questions.
Yeah. Thanks, Valentina. Very nice presentation. A lot of work going on for the next generation dendrochronology.